Hi friends, let us discuss about the cell wall in this session. The term cell wall denotes something which protects the cell. Wall means barrier. So cell wall is the outermost covering that protects the cell from the various atmospheric distortion. Okay. In order to just for better understanding, see fruits are having their own peels. For example, the orange pulp is protected uh, by its covering, by its own skin. Even human beings are covered with a skin, with a layer of skin, which is also made up of cells, like epidermal cells. Okay, now we are seeing in detail about a single cell. A single cell, when it is covered with its outermost covering, in order to protect from the various atmospheric conditions, it is called as cell wall. So, do these cell wall are these cell wall present in all the cells? Do all cells have the cell wall? No. Animal cells doesn't contain the cell wall. Cell wall is present in plant cells, bacterial cells, fungal cells, whereas the animal cells lack cell wall. Okay, it is a non-living layer, and cell wall is made up of various components in various type of cells. Like in plants, it is made up of cellulose. In bacterial cells, made up of peptidoglycan. Fungal cells made up of chitins, glucan, mannans, etc. So this cell wall supports, protects, gives shape to the cell, and it is the outermost portion, and next to the cell wall is the cell membrane. I said before that bacteria contain cell wall, but there are few exceptions. It is just a general knowledge that you should be knowing. So Halobacterium, Halococcus, Mycoplasma, these bacteria and all are devoid of the cell wall. Okay, it is just a GK. So the picture beneath depicts the bacteria Mycoplasma. They are the colonies of Mycoplasma. Colony is something, a single bacteria multiplies, divides, and multiplies into many millions and trillions of bacteria. Okay, they form a single colony, which appears like as a dot up to these colonies will be up to 2 mm. Okay, so a single bacteria cannot be observed under microscope. Cannot be observed through the naked eye, whereas colonies can be observed through the naked eye. Okay, this you should be knowing. This picture is the Mycoplasma colonies. Mycoplasma is a bacteria which causes lung infection, and it results in the pneumonia, mycoplasmic pneumonia. Okay, these are mycoplasma. When mycoplasma colonies, when observed under microscope, they give the appearance of fried egg, isn't it? They are like fried egg, right? Like center portion having a yolk sac and all. The periphery is like a consisting of albumin. Hmm? So they determine the shape. The cell wall determines the shape of the organism. How come? The bacteria consist of various shapes. They are of various shapes. They are cocci. They are called cocci if they are spherical. They are called rod shape. There are spiral bacteria. All these are how the particular bacteria is having the particular shape. It is because of the cell wall. If there is no cell wall, the shapes couldn't be determined. Okay, based on the shapes also, that is based on the morphology also, the microscopic, laboratory diagnosis are done. So cell wall is a very important. It holds the cells together and it prevents the cells from bursting due to osmosis, or etc. So we'll see in detail about the bacterial cell wall. Bacterial cell wall is made up of peptidoglycan. Okay, peptido means peptide. Peptide means amino acids. Group of amino acids together linked to form peptide chain. Okay, glycan means sugar. Okay, amino acids and sugar molecules together is called peptidoglycan. This peptidoglycan is the forms the I mean is made up of the cell wall is made up of the peptidoglycan, and based on the quantity of the peptidoglycan, they are classified as thick, gram positive and gram negative. Gram positive bacteria have thick peptidoglycan layer, whereas gram negative bacteria contain thin peptidoglycan layer. Based on the amount of peptidoglycan layer, they are classified as gram positive, gram negative. Okay, so gram positive bacteria and gram negative bacteria both contain peptidoglycan. Hmm? What is peptidoglycan made up of? They are made up of two alternating series, that is two subunits NAM and NAG. They are arranged in alternating series. NAM is N-acetyl-muramic acid, NAG is N-acetyl-glucosamine. These both are arranged in alternating series, to which uh, uh, they are joined to form a glycan chain. To each NAM molecule, 4 amino acids. 4 means tetra amino acids. When group of amino acids, peptide. Okay. To each NAM molecule, 4 tetrapeptide chain is attached. Okay. So NAM NAG, NAM NAG, NAM NAG. Likewise, it is alternatingly arranged with each NAM having tetrapeptide chain and results in the formation of a glycan chain. Okay. And this high molecular weight linear polymer serves as a backbone to peptidoglycan. See, this NAG, NAM, NAG, NAM. This part A picture is the zoomed picture of part B. Okay, NAM, NAG, NAM, NAG. This NAM is having a tetrapeptide chain. Likewise, the another glycan chain is having a tetrapeptide chain. These adjacent glycan chains are linked together by a peptide interbridge in gram-positive cells, whereas in gram-negative bacteria, they are linked directly without the help of a peptide interbridge. Okay, teichoic acid. It is found only in the gram-positive bacteria. It is a it is the linkage between the cell wall and the cell membrane. Okay, this teichoic acid is either made up of ribitol phosphate or glycerol phosphate with various sugars and D-alanine. Okay. This teichoic acid binds with the NAM of the peptidoglycan. Okay, it connects the cell wall with the plasma membrane. Lipoteichoic acid is something which is linked to the plasma membrane, that is lipoteichoic acid lipid portion of the plasma membrane. You'll get a better understanding in this picture. See, teichoic acid is attached to the NAM of the peptidoglycan layer, whereas lipoteichoic acid is attached to the NAM of the peptidoglycan layer and to the uh, pep peptide peptide region of the plasma membrane. Okay. Gram-negative bacteria, in addition to the peptidoglycan layer, they have a lipopolysaccharide. Okay. In addition to peptidoglycan layer, they have peptidoglycan, but they have very thin peptidoglycan layer. In addition to peptidoglycan, they have a lipopolysaccharide layer, which is made up of O polysaccharide and lipidae. Lipidae is the portion of lipopolysaccharide which is present in the lipid bilayer. It is more toxic and small amounts introduced to produce immunity. So how is immunity produced? In vaccinations and all, either the cellular components or the whole bacteria or viral cell, either in the inactivated or attenuated or killed forms, are suspended in order to produce the immunity. In some types of vaccines, this lipid A portion is a small amount of this lipid A portion of the cell wall is subjected to use as some vaccines to produce the immunity. Okay. And the O polysaccharide is a portion of LPS that is present outside the membrane. See, it is present outside the membrane and it is made up of a chain of sugar molecules. Also, it has many proteins called porins, etc. And this periplasm is the fluid filled gel like fluid between the outer membrane and the plasma membrane, and all the secreted proteins are present in the periplasm. So just recall, based on the amount of peptidoglycan present, they are classified as gram positive, gram negative. Gram positive bacteria contains thick peptidoglycan, whereas gram negative bacterial cell wall contains thin peptidoglycan. Gram positive cells contain teichoic acid, lipoteichoic acid. Gram negative cells are devoid of teichoic acid, lipoteichoic acid. Gram positive organisms doesn't contain LPS, whereas gram negative organism contains lipopolysaccharide. Altogether, the cell wall is made up of peptidoglycan in the bacterial cells, whereas in plant cells it is made up of cellulose. In animals, the cell wall is animal cells lacks the cell wall, and fungal cells it is made up of chitin, glucan, mannans, etc. Hope you like this session. Um, if you really liked my video, please do subscribe. Thank you.